Okay, this is going to be the final section in 7. This is 7D and DNA technology. So we're going to start with genetic engineering. Genetic engineering is anytime we take a critter and we want to change it for some reason. We're changing or alterating the DNA in the organism, and what we really do is we go in and we snip out sections and we either tie it up or we replace it with sections of maybe phenotypical traits that we like. Taller, furrier, shinier, insect resistant, whatever we want. Here's an example of a lady, Thanksgiving, preparing dinner, I guess, and she says, Isn't genetic engineering amazing? 2 years ago, who would have even imagined such a thing as a turkipede? And if you take a look, hopefully you like legs. And you can do other neat things, like you can have apples that taste like watermelon, or you can have apples that taste like peaches. All you got to do is figure out what part of the DNA an apple is that makes an apple taste like an apple, figure out what the same part is in a peach, and just snip it out, replace it, and you got it. DNA fingerprinting. DNA fingerprinting is a method which we actually use to identify the DNA banding for a unique individual, and they actually say it's more individual than a fingerprint. I'm not sure if that's true or not, but this is a DNA sample taken from an individual, and you can see all these little lines that are on there. The doctor's holding it up, and you see all these little lines. They put the DNA in liquids. The liquids actually dissolve the material, and as the material rises up uh, through the material, it actually leaves the DNA marks behind it. So, who committed a crime? This is the material that's actually taken: blood, semen, saliva, skin, hair, DNA sample that was left at a crime scene. This is suspect 1, suspect 2, and suspect 3, and who actually committed this crime. And all you have to do is just look at the lines in the crime scene, take a look at the suspects, see who's got the same lines, and I think it's going to be, I don't know where, it's going to be suspect 2. You can also use it for like who's your daddy. And in this one, we actually take a look at mom and dad, mom and dad, mom and dad, and here's a baby. And then we look at the lines the baby has, and we have to find those in the parent. Now, we can actually have more in the parents because mom got it from mom and dad and may actually only give part of the stuff she received from mom and part of the stuff she received from dad, so it may actually be a little different. But if you take a look, um, doesn't look like this can be the father. Almost nothing lines up. Probably the same thing with the mother. This father does actually, it looks like it's got some of the material, and if the father doesn't have it, does the mom? And if you match it up, it actually looks like that's the mother and the father. The Human Genome Project. This was a study actually conducted by the United States government as well as private business, and what they did is they took the 23 chromosomes of the 46 pairs, the 23 chromosomes, and actually started looking at all the A's and the T's and the C's and the G's, and actually figured out which ones actually worked with genes, and came out with a map. And this is a sequence or a list of all the genes inside of a human genome. And we found out that we have somewhere between 20 and 30,000 genes, and all of those wrapped up on 46, really 23 chromosomes. This is one of the chromosomes. This is actually chromosome 1. And you can see this individual chromosome has 263 million bases. So 263 million A's, T's, C's, and G's. And you can see it actually causes problems. It also provides traits. So cataracts is on this one, and prostate cancer, and brain cancer, and actually syndromes, and suspensions, and type 6 syndromes, and all kinds of uh, material. But these all code for proteins, and the proteins are either read correctly or they're not read correctly. And we may do an activity with the 23 chromosomes. Transgenic organisms, these are also known as GMOs, or genetically modified organisms. And this is when we take an organism who has material and we alter it somehow using genetic engineering, cutting and splicing. So let's say we have corn, and this corn's actually being infected by a critter called a European corn borer. And if you've ever seen people actually opening up corn at the store, they're actually looking for this type of material, as well as maybe full ripening. But all you really have to do is just cut this stuff out and the corn is fine, although lots of people don't want to do that. But we found a bacillus that actually has the ability to get into this borer and kill it. So why couldn't we take the information that actually makes that? So here's the BT gene. This is the part that actually does the killing of the borer. Here's a corn DNA. Why don't we slice out a piece of corn DNA, slice out the BT gene from the bacillus, and actually take that and splice it inside the corn. So basically any part of the corn that's out there, those are the ears, but any part of this corn that this thing actually eats, it gets this poison and it kills the borer. Wouldn't that make sense? Problem is that's a GMO raised corn, and a lot of people actually say that this corn is not natural and we shouldn't be giving it to people to ingest. It's fine to give to chickens, it's fine to give to pigs, it's fine to give to cattle or horses, and then we go ahead and eat some of those. But apparently it's not good to eat the corn, but it's okay to eat the critter. You can also do the same sort of thing if you take like a firefly and you take the ability of a firefly and you take that part of the genetics out and you go to a plant and take another part out, you could actually end up with glow-in-the-dark plants. Or why stop there? Why not even get glow-in-the-dark mice? Pretty cool, neon green. Then we have something called cloning. Cloning describes a number of different processes we can use to produce genetically identical copies of an entity. So it could be you, it could be a dog, it could be an elephant, it could be a mouse, it could be food, it could be anything, but you're making an genetically identical critter. This copies material which has the same genetic makeup from the original and actually is referred to as a clone. Here's an example where we actually have a human mother and another human mother, and they both donate eggs. And the egg gets the nucleus removed, and then we take the outside of this egg, the membrane, and we actually fuse it with the nucleus of this woman. And why don't we just take this and implant it into this mom? Well, the outside of this cell, mom would know is not hers, and it would kill it, it would lyse it. So what we do is we actually fuse it. So now we actually have this mom's nucleus inside this mom's egg, and then we let it grow for a while, and we let it multiply, go through mitosis and double and maybe triple in size, and then we implant 1, 2, or 3 of these inside of this mom, and she actually is the surrogate mother. The baby grows through those 42 mitotic cycles inside of this woman, even though it's genetically the same as this mom. 1996, the world was actually introduced to Dolly. Dolly is a lamb and actually looks very much like the mom. Problem is Dolly actually died within the first year, so there were some other issues that we were not aware. But we could do that with basically any kind of critter. Imagine if we did that with you. So this is what we did with Dolly. So Dolly was a Finn Dorset ewe, and what we did is we took eggs out of Dolly's mom, and we actually put them inside of a container that had low nutrient culture, and then we actually took the DNA from that cell and we fused it inside of donor egg from this side, where we actually took a micropipette, really small pipette, sucked out the nucleus. Then we took this DNA and actually the shell and the DNA, and we infused it inside of this cell membrane, and then we put electric pulses in it, and we fertilized it, and we let it grow through a couple mitotic divisions, and then we took this and we implanted inside of this surrogate mother, who was a Scottish blackface ewe, and she gave birth to Dolly, genetically the same as this mom. So we took cells, implanted into an egg from another ewe, and then let that mom um, feed and nurture the developing embryo, and then gave birth to Dolly, the sheep. If you want, you can actually go over and look at this NPR. You can clone your dog. What you'll find out is they'll tell you can clone your dog if you want. It's a South Korean company. All you got to do is come up with $100,000, and you can have two of the same dogs. DNA repair. If you remember, we have a cell cycle. We go through the mitotic division right here, and then we end up with one set of chromosomes. We go through and we synthesize, making two sets. It splits up, and then it makes another copy, and then we end up with G2 here. This is where the cell grows. This is where the synthesis, where it creates another string of DNA, and this is where it actually checks, makes sure everything is done correctly, has all the right organelles, has the right size, has the DNA, and the DNA is done correctly, and then it lets it go through another mitotic division to make more cells. And again, it's all making up these proteins. So we got to make sure we make these proteins correctly. And then I think the last slide, this is actually Al's foreign and domestic DNA repair. Another teacher actually took it and put their name on it. I can't find the original, but this one is out on the web. And it says, boy, you're lucky. Nobody was injured. Your base pairs are all out of alignment, and your reading frames all messed up. So if you take a look, it looks like that DNA's got some problems, and he says he can go in there and he can fix it. This is really the last slide, and this, if you take these things and go into, and I'll put the links to this video, if you go into YouTube and you type flu attack, how the virus invades your body, or better yet, the inner life of a cell, you can see two really cool images. And this is a microtubule, and we have a protein that actually walks like a person, and it's dragging a vacuole. You can see another microtubule up there, and there's another one right there, and another vacuole being drugged by another protein. But this is the way cells work, and we have all kinds of things that help identify and help to attach to other organisms. And this is actually from the virus video, and how we actually get colds, how it actually causes damage, and how our body fights the cold and makes sure that the cold doesn't end up killing you. Okay, that's it. Thank you. Bye.